In this lecture, we're going to talk about osmosis and diffusion. Diffusion is any movement of molecules from an area of high concentration to an area of lower concentration. So in this diagram, you can see like food coloring or dye that has been dropped into the water. When you drop it into the water initially, it does not spread out very quickly. But if you wait and don't stir it, eventually those molecules will move from an area where they are highly concentrated, and they will move out to areas where they are lower concentrated, and eventually they will fill the space evenly. And so that is diffusion. Osmosis is very similar to diffusion, but in order for osmosis to occur, it has to travel through a semi-permeable membrane. And so in this case, you can see that the water on this side, represented by red dots, is diffusing through the membrane, and the water is pulled in that direction because of osmotic pressure. It wants to dilute the high concentration solute. The word permeable is just a word that means that it allows substances to pass through. So the cell membranes on all of our cells are permeable. You could also think about it like a window screen on your house. When you open the glass window so that it's just the screen, it allows some things like air and dust particles and things like that to go through, but it keeps other things out of your house, like large insects or birds and things like that. If something is impermeable, then it does not allow things to pass through. There are three types of solutions that we call osmotic solutions, and these are solutions that cause water to move across a membrane because of a concentration gradient. So a hypertonic solution occurs when you have a solute out, which is a liquid that has something dissolved in it. If the solute concentration of a solution is higher on the outside of the cell than on the inside of a cell, then it's hypertonic. It means that there's more dissolved particles outside of the cell than inside of the cell. The word hyper means more, which is why the hypertonic solution has more particles. And water in this type of solution will move out of the cell and into the solution on the outside of the cell. When a cell is in a hypertonic solution, the cell shrinks or gets smaller because the water wants to leave the cell to dilute the solution that is outside of the cell. So in this diagram, it shows a hypertonic solution. You can see that the ratio of water to dissolved particles inside of the cells is lower than the ratio of water to dissolved particles outside of the cell. So a cell in a hypertonic solution loses water. You can see there's arrows here showing the direction of water movement. The water leaves the cell and it causes the cell to shrink or get smaller. This is what a plant cell called Elodea looks like in a hypertonic solution. You can see in this diagram the cell wall doesn't move because you can see the cell wall is still rigid, but you can also then see the cell membrane, which is here, has pulled away from the cell wall because water has rushed out of that cell. When water rushes out of a cell like this Elodea cell, it's called plasmolysis. Here's another diagram of a cell that is in a hypertonic solution. It is losing water. This happens to be a red blood cell, so you've got lots of those in your body. And if they're in a solution that is hypertonic, this is what they look like. They normally look kind of like donuts. A hypotonic solution is opposite. It has a lower solution or solute content than the fluid that's inside of a cell. So there's less dissolved particles outside of the cell than inside of the cell. Hypo means less, so it makes sense that there's less dissolved particles or fewer dissolved particles. When a cell is sitting in a hypotonic solution, water moves into the cell and it can cause the cell to swell, and if it swells too much, it might burst and it can kill the cell. So in this diagram, you can see the hypotonic solution causes water to rush into the cell because there is less dissolved solute outside of the cell than there is inside of the cell, so the water wants to rush into the cell to dilute it. Here you can see red blood cells again, and these are red blood cells that are in a hypotonic solution. So you can see some of them, like this one, has lost its characteristic depression where they have that concave cavity, and it's swelling up because it's taking on water. Water will always move towards a more concentrated solution to dilute it. So if you had, for example, a U-shaped test tube with a semi-permeable barrier, and you had a low concentration solution on one side of the barrier and a high concentration of the solution on the other side, water would move into the hypertonic solution, the one with more dissolved particles or more solute. And then that will change the volume of the two different sides of the tube. So you can see here the water level went down, here the water level went up. The last type of osmotic solution is an isotonic solution. It's when the solute concentration of the solution is equal to that is in the cell. So this means that there's the same amount of dissolved particles inside of the cell as there are outside of the cell, which means there's no net water movement. So the cell does not shrink or swell. It stays the same. So to review, water always moves towards the area that has a higher concentration of dissolved solute. And so if you have a cell that is in an isotonic solution, the same amount of water will go into the cell as comes out of the cell, and the cell does not change sizes. In a hypotonic solution, the water rushes into the cell because there's more dissolved solute inside of the cell. This causes the cell to swell. And finally, in a hypertonic solution, there's more dissolved solute outside of the cell, so this pulls water out of the cell and causes the cell to shrink. In this diagram, you can see all of the different effects of these solutions on blood cells. So you can see in a hypotonic solution, the cells have swollen up and do not have their characteristic donut shape of a normal blood cell. In a hypertonic solution, you can see that the cells have shrank considerably and have taken on a completely different texture. And in an isotonic solution, you have what a classic blood cell looks like. They're concave, so a slight depression in the middle, and otherwise generally quite a round cell. Here you get to answer the question, what kind of solution is this? So a hint, you can see there's 10% salt solution outside the cell, 10% salt solution inside of the cell, and it tells us that water goes in both directions. So go ahead and decide if you think if that cell is hypertonic, hypotonic, or isotonic. Here we're going to ask the same question. What kind of solution is this? So details you should pay attention to. You have a 10% salt solution outside of the cell, a 20% salt solution inside of the cell, and here it says that water moves into the cell. So then you can decide if you think that the solution outside of the cell is hypertonic, hypotonic, or isotonic. The last question is the same question as before, but we have new solutions. So on the outside of the cell, we have a 20% salt solution. Inside of the cell, we have a 10% salt solution. And here it tells us that water moves out of the cell. So you get to decide one more time if you think the solution outside of the cell is hypertonic, hypotonic, or isotonic.